Okay. So if we are looking for structure of Anatropus ovule, what we going to be observe? Okay. डायग्राम मध्ये स्ट्रक्चर एकदम व्यवस्थित आपल्याला कळेल बघा. Okay. So here we are having the complete structure of ovule. So in this case, okay, in this case we're going to be observe that here is the placenta. Okay, this is the placenta. Placenta is connected to the ovule by the stalk-like structure called funicle. So by the funicle, ovule attaches to the placenta at the site of hilum. So this is the hilum site through which ovule is going to be attached to the placenta. Then complete ovule carries the two layer, outer integument, inner integument. Okay, then this is the base. Okay, this is the base. This is the apex. So at the apex, we're going to be observe the one opening called as micropyle. Okay, micropyle. So at the micropyle side, we're going to be observe nucellus. Okay, nucellus, and it is going to be generate the three different cell. Two are the synergids and one is the egg. Okay, two synergid and one is the egg. So synergid look like same. Okay, इथे एक nucleus है, इथे एक nucleus है. So these two are the synergid, and central we have egg. Then here embryo sac carries the secondary nucleus, which is diploid in nature. And then here we have the antipodals, which are 3 in number. And whatever the base of ovule called as chalaza. Okay, base la apan म्हणतो chalaza. Okay, is the structure. So let's come back to the point. Okay, so each ovule develop inside the ovary and is attached to the placenta by stalk-like structure called as funicle. The place of attachment of funicles with the main body of ovule is called as hilum. In angiosperm, the most common types of the ovule is called Anatropus, in which micropyle is directed downward. Okay, म्हणजे apex site ला micropyle, and this is present adjacent to the funicle. म्हणजे ज्या ठिकाणी ovule placenta ला connect होतोय, on that side we going to be observe the micropyle. Then the ovule consists of central parenchymatous tissue and nucellus, which is surrounded by usually two protective covering. आपण integument म्हणतो त्याला. So it includes the outer integument and inner integument. So whatever they are inside, the all cells are parenchymatous in nature, and all these cells are surrounded by protective covering called integument. Okay, we call this as an integument, outer and inner integument. A narrow opening at apex of ovule is called as micropyle. Okay, we also call another site, whatever the opposite to the micropyle, we're going to be observe base called as chalaza. Okay, and embryo sac, female gametophyte, is oval multicellular structure embedded in the nucellus. So inside the nucellus, we're going to be observe the total 6 cell. 6 cell आपल्याला दिसतात. So it is multicellular structure. त्या 6 cell आपण पाहिलेले आहेत. Okay, these are the 7 cells. 1, 2, 3, 4, 5, 6, 7. Okay, अशा 7 cell आपल्याला पाहायला मिळतात. Okay, let's move to the next one. How exactly ovule is going to be formed? Female gametophyte is going to be formed. So let's see. So process is initiated with the meiosis of mother cell. So mother cell is the diploid. It is going to be proceed for meiosis. So we know that meiosis is the reductional cell division. So first process in which the two cells are going to be formed by mitotic like function, and later on this is converted into the haploid 4 cells. So this is the meiosis, reductional cell division, convert the genetic material to N to N. Here we are having the 4 cells which is going to be formed from the megaspore, mother cell, diploid cell. The cell are going to be observed that whatever the cell is in turn remains as it is, and remaining 1, 2 and 3 cell are going to be degenerate. ह्या cell degenerate झाले. Okay, and the one cell is going to be increase their size. म्हणजे यामधला सायटोप्लाझ्मिक कंटेंट जो आहे, this cytoplasmic content is added to the 4th cell. So megaspore or embryo sac, it is going to be formed. Haploid, these are the haploid cells. So growth and nourishment of nucellus, it is going to be form, increase in the size. It is going to be proceed for mitosis. So at the nearest to the end of micropyle, आपण पाहिलं की micropyle is the opening present at the apex side where the funicle is going to be joined to the placenta, where this cell is going to mitosize and it is going to be produces 2 nucleus. 2 nucleus तयार झाले सेलच्या आतमध्ये. Again, it is going to be proceed for mitosis. It is going to be produces 4 cells. Later on, this 4 nucleus is again going to be divided. 4 चे 8 होणार. त्यापैकी 1, 2, 3. It is going to be form 3 antipodal cells. Then 2 are present at the center. Okay, and remaining 3 which is present at here, but it's not look like. त्यापैकी बाहेरच्या ज्या 2 आहेत, these are 2 synergids. And in the center, whatever this present, it is the egg. So like this way, 8 cell is going to be formed. 8 cell तयार होणार. 1, 2, 3, 3 antipodal cell. Then 2 egg female gamete which is at center, and then 3 which include the 2 synergid and 1 egg gamete. Then it is going to be proceed for the nuclear fusion. Whenever the nuclear fusion is going to be occur, these 2 cells are going to be joined with each other. It is going to be formed single cell. So this cell is diploid. This cell is not diploid. So this is the mature embryo which is going to be carry 3 antipodal cell, the single cell at the center which is diploid, then 2 synergid and 1 egg cell. अशा total 1, 2, 3, 4, 5, 6 आणि 7 सेल तयार होणार की ज्यामध्ये एक जी आहे ती डिप्लॉइड आहे and remaining all 6 are haploid. ते इथे सांगितलेलं आहे मॅच्युअर एम्ब्रियो सॅक ऑफ फिमेल गॅमेटोफाइट just before the fertilization carry 7 nuclei which is going to be show the 6 are haploid and 1 is diploid. Okay, so this is the way. परत एकदा रिपीट करतो बघा मी. So what is going to be happen? So megaspore or mother diploid cell is proceed for the meiosis, and after meiosis it is going to be form 4 cell. आपल्याला माहिती आहे की 4 cell कशा meiosis मध्ये तयार होतात ते. Then out of these 4 cell 1, 2, 3 are degenerate. It is going to be add the cytoplasmic content into the single cell. Just साईराज घाडी जरा म्यूट कर, म्यूट होत नाही. साईराज घाडी म्यूट करा. Okay, so let's see. Degenerate the 3 cell, and after degeneration of the 3 cell, it is going to be increases the cytoplasmic content of single cell. Then it is going to be proceed for the growth and nourished by the nucellus. It is going to be proceed for the mitosis, and it is going to be form the larger 2 cell or 2 nucleus. Then again it is going to be proceed for mitosis to produce 4 nucleus. Again it is going to be divided to produce 8 nucleus. Then here we are having the 3 antipodal cells. Then 2 nuclei at here, 2 synergids at here, and 1 egg cell. Later on, these 2 cells are joined with each other to produce the diploid cell. So 6 are haploid cell and 1 is diploid cell is going to be formed. Let's move to the next one. Okay. Then we are having the megasporogenesis. So megasporogenesis, it is the process of formation of haploid megaspore from the diploid megaspore mother cell. So megaspore mother cell, it is the cell which is after the meiotic division, it is going to be formed ovule. So megaspore mother cell become distinguished from nucellus more or less in the center, but towards the micropylar end of ovule. Means whatever the megaspore is present, it is distinguished in the nucellus, but it is away from the somewhat center. म्हणजे center पासून थोडंसं बाहेर गेलेलं असतं. Okay, micropylar end or ovule. So development of female gametophyte, megaspore mother cell undergo meiosis to form the linear tetrad of haploid cell. Okay, megaspore mother cell is going to divide and going to be produces linear tetrad. आपण पाहिलं की 4 tetrad cell तयार होतात. So upper 3 cells of megaspore abort and lowest one towards the center of nucellus is remains. म्हणजे आपण पाहिलं बघा 1 2 3 आणि 4 असे 4 सेल आहेत. त्यापैकी 1 2 आणि 3 abort झाले. And this is going to be reached towards the center of nucellus and it is going to be functional. ही जी आहे ती सिंगल फंक्शनल आहे. It acts as an first cell of the female gametophyte and generally one megaspore towards the center is functional megaspore and it is in fact the first cell of female gametophyte. It undergo the 3 successive cell division. So pre-nuclear mitotic division. So this is going to be proceed for total किती 3 successive cell division होणार आहे. So in the first division एकाचे 2 झाले, in the second division 2 चे 4 झाले and in the third division 4 चे 8 झाले. हे आपण पाहिलेलं आहे मागच्या डायग्राम मध्ये, लक्षात घ्या. Okay, so it is going to be form total 8 cell. But these 8 cell is going to be generated by mitotic division. Thus total 8 nuclei are formed, 4 of which are located at the pole. मग यापैकी वरती 1 2 3 खाली 1 2 3 असं होणार. At each pole the one nucleus from each pole migrate. Okay, इथे 4 असतात आणि इथे 4 असतात दोन पोलला. So one of them is going to be migrate. This one is going to be migrate at center here. This one is going to be migrate at here. मग इथं 3 राहिल्या and here we remains 3. मध्ये 2 झाले. So these two are going to be joined and it is going to be produces the diploid cell. One nucleus from each pole migrate towards the center are called as polar nuclei. ह्या दोन ज्या आहेत त्यांना आपण polar nuclei आहे. ह्या दोनना synergy हा एक सेल आणि दिस इज द अँटीपोडल 3 सेल्स. अशा सर्व सेल आहेत. मागची डायग्राम जी पाहिली त्याचंच एक्सप्लेनेशन आहे. Then the 3 nuclei towards the micropylar end consist egg apparatus. Okay, egg apparatus. Central is the egg cell. Consist of the large central haploid egg cell and 2 supporting haploid synergid cell. Okay, मगाशी आपण पाहिलं की मायक्रोपायलर एंडला सेंट्रल सेल जी आहे ती लार्जर अँड 2 ज्या सेल आहेत त्या सपोर्टिव्ह सेल आहे. सो दिस इज गोइंग टू बी मेक synergid. Synergid shows hair like projection called filiform apparatus and which guide the pollen tube towards the egg. Then antipodal cells are group of 3 cells present at the chelazal end. The 2 haploid polar nuclei of large central cell fuse to form diploid secondary nucleus. This whatever the cells which are migrated to the center, these are going to be 2 haploid polar nuclei of large central cell fused to form diploid secondary nucleus, की ज्याला आपण definite nucleus असं म्हणतो, just prior to the fertilization. So what we going to be observe in the cell, we going to be observe that the 7-cell and 8 nucleated structure. 7-cell and 8 nucleated structure present in the embryo sac. So out of these, the 6 are the haploid and 1 is the diploid. त्यामुळे आपण म्हणतो की 7 cell and 8 nucleated structure. So this method of the embryo sac development form a single megaspore is described as monosporic development, and in angiosperms the development of female gametophyte is endosporous. Okay, endosporous, that is within the megaspore development of female gamete is going to be occur. So female gametophyte is colorless, endosporic, and it is concealed in the ovule enclosed by the ovary. Okay, it is going to be concealed in the ovule enclosed by the ovary. So this is the monosporic development. Let's move to the next one. Okay, then second part of the plant reproductive include the pollination. Okay, इथून पार्ट चेंज होते आपण आतापर्यंत पाहिलं की फर्टिलायझेशन कसं होते, how exactly pollen grains and ovules are going to be formed. ह्याच्या पुढचा पार्ट येतो तो म्हणजे pollination process, how exactly pollination process is going to be occur. So pollen grains being non-motile, angiosperm have evolved the strategy to use the abiotic agent like the wind and water, and biotic agent like bird, insect, snail to carry out the pollination, because pollen grains are non-motile, it's not having the abilities to travel. So whatever the pollen grains present in the one flower, it is going to be transmitted to the other by abiotic agent wind, water, biotic agent bird, insect, snail. So feeding the visitors and exploiting their mobility for pollination and seed dispersal. So two things are going to be happening. Angiosperm going to feed bird, insect, snail, and whatever the mobility of this bird, insect, and snail, they utilize for pollination and for seed dispersal. So pollen grains are non-motile and they are usually carried from the flower to flower by means of external agent. Pollination is transfer of the pollen grains from anther to the stigma of the flower. आपल्याला माहित आहे की anther is the androecium, male reproductive structure. Stigma is the part of gynoecium. This is female reproductive structure. Anther or pollen grain from anther to stigma carried by the several pollinating agent. So it is the prerequisite for the fertilization. For the fertilization, we need to transfer the anther to the stigma. Okay. So both male and female gamete are non-motile. So moreover, gametes are produced at two different sites. In the self-pollination, in the self-pollination is a type of pollination which occur in the single flower or two flowers on the single plant. त्याला आपण म्हणतो self-pollination. So pollination which is occur between the single flower and two flowers of the single plant, we call this as an pollination. त्याला आपण self-pollination. Then when self-pollination is going to be occur, it will be leads selfing, inbreeding, because the both flower belongs to the same plant. Flower is single, belongs to the same plant. So in turn, it is going to be leads inbreeding. It's not going to be change any genetic constituent की ज्याला आपण selfing म्हणतो. In contrast, if we are looking for the cross pollination, transfer of the pollen grains from anther of one flower to the stigma of another flower of different plant, it will be leads cross pollination. So cross pollination is in turn going to carry out outbreeding, change the genetic constituent. But pollination can be further divided into the three types on the basis of source of pollination. Pollinationचा source काय आहे? त्या basis वरती again the pollination divides into the different type. First one, autogamy. So auto means cell, gamy means gamete, self pollination. So this is the type of pollination in which bisexual flower. So bisexual flower means what? Flower which is going to be carry both reproductive structure, androecium as well as gynoecium. त्याला आपण bisexual flower is pollinated by its own pollen grains. Offerings are genetically identical to their parent. For example, pea plant going to be carried out autogamy. Then Xenogamy. Xenogamy means what? That it is the transfer of pollen grains to the stigma of different flower produced on the same plant. Means flower is unisexual. म्हणजे हे मेल फ्लावर आहे, सॉरी फिमेल. This is the male flower. But these two flowers are present on the same plant. हे दोन्ही फ्लावर सेम प्लांट वरती आहे. So if pollen grains from here transfer to here, we call this as an Xenogamy. The transfer of pollen grains to the stigma of different flower produced on the same plant. Okay, functionally these both flowers are generated from the same type of the cell. Because of this, again it is not going to be bring any genetic variation. का? कारण हे पूर्ण plant सेमच आहे म्हणून. So, but it is going to be show ecological significance. Cucurbita maxima, it is similar to the Antogamy, as pollen grains come from the same plant. Let's move to the next one. Okay. Then we are having last one, Xenogamy. So Xenogamy means what? So it's include the cross pollination, it include the outbreeding. So it is the type of cross pollination when pollen grains of one flower deposited on the stigma of flower of different plant belongs to the same species. With the help of pollinating agency, it generates genetically varied offspring. म्हणजे काय झालेलं आहे? This is the one plant, or this is the one flower. Okay, flower of one plant. So here we are having the flower of different type, or this is the also different plant. So whenever pollen grains from these carried out pollination at here, it will be leads cross pollination. त्याला आपण म्हणतो Xenogamy. So Xenogamy, because it is occur into the two different plant, it will be generate genetically varied offspring. त्याला आपण cross pollination and outbreeding असं म्हणतो. So majority of flowering plant depends on their transfer of pollen grains. Is going to be show different types of the pollination. Virtually all seed plants need to be pollinated, and most of the food fiber crop grown throughout the world depends on pollinator for pollination. म्हणजे food आणि fibers crop जे आहेत, those are totally depends on pollinators for their reproduction. Then on the basis of agent responsible for pollination, काही categories आहेत. त्यामध्ये first one abiotic agent, second one biotic agent. So let's see. Abiotic agent. These are non-living agents which are include the wind and water, which is going to be carried out the pollination. So let's see. काय? First one, pollination by wind, also called as anemophily. So most of the important crop plant are wind pollinated. So these are include wheat, rice, corn, rye, barley, oat. हे सर्वच्या सर्व plant जे आहेत, those are along with the palms are going to be carried out wind pollination. So what type of the adaptation observed with the anemophilic flower? The flowers are small and it is the inconspicuous. Okay, inconspicuous म्हणजे pack नाही, colorless, because it's not going to be attract the pollinating agent. It's not carry the nectar because it is going to be carried out transfer of the pollen grains with wind. Then the fragrance and odor is not present in them. The pollen grains are lightweight because we need to transport it with the wind, dry and produce in the large number to increase the chances of pollination. Then stigma is feathery to trap the pollen carried by the wind. Stamens are exerted with the long filament and versatile anther. Okay, versatile anther hai, long filament hai, so that it is going to be transmit the pollen grains to longer distance. Stamens and stigmas are exposed to the air current. पुढे गेल्यानंतर चित्र बघितल्यानंतर तुम्हाला काय हे आहे. So this is the example of the maize. काय काय characteristic दिसतात बघा. So this is the inflorescence. So in the inflorescence we are going to be observed stamens are long, so that whatever the pollen grains, it is going to be transmit with your longer distance. Then here we are having the flag leaf. Okay, flag leaf. Okay, then tassels internode. Okay, internode. Then here we are having the female reproductive structure, female inflorescence ki ज्यामध्ये आहे style. Okay, style. So it is going to be catch for the pollen grains. So here we have the leaf, stem, prop root and root. Okay, like this one, it is going to be generate the particular structure. मग ह्याच्यामध्ये मेज मध्ये काय काय कॅरेक्टरिस्टिक्स आपल्याला दिसतात त्या कॅरेक्टरिस्टिक आपण थोड्याशा पाहिल्या इथे. परत एकदा एकदम फास्ट मध्ये रिपीट करतो. सो व्हॉट एव्हर द फ्लॉवर्स आर स्मॉल इनकॉन्स्पिक्युअस म्हणजे पॅक नाही, कलरलेस, विदाउट नेक्टर, विदाउट फ्रॅग्रन्स. पोलन ग्रेन्स आर लाइट वेट, ड्राय, प्रोड्यूस इन द लार्ज नंबर टू इंक्रीज द chances of pollination considering the wastage of pollen grains. Stigma is feathery, trap the pollen carried by the wind current. Stamens are exerted with the long filament and versatile anther, so that anther is going to be transferred to the far distance. Then stamens and stigma are exposed to the air current. So we're going to be observe it with this example. Then second one is the hydrophily, anemophily pyramid, the pollination which is going to be occur by the ear. Now we are having the hydrophily. So hydrophily means what? It is nothing but the pollination which is going to be occur by water. Okay, we're going to be observe that 30 genera of aquatic monocot, including the Vallisneria, Zostera, and Keratophyllum. These all genera is going to be carried out pollination with water. Adaptation काय काय बघ. Flower are small, inconspicuous. Perianth and other floral part are unwettable. पाण्यामध्ये त्यामुळे लगेच it's not going to be captured the water. Pollen grains are long, unwettable due to the presence of mucilage on their surface. Nectar and fragrance are lack. नहीं. Hydrophily is the of two type. First one is the hydrophily. Okay, sorry, hypohydrophily. So hypo means below the average. Then आपण hypohydrophily म्हणतो. So pollination occurs below the surface of water. So we call this as an hypohydrophily. Okay, here pollen grains are heavier than water. So sink down and caught by the stigma of female flower. For example, sea grass, की ज्याच्यामध्ये pollen grains are going to submerge into the water. Okay, below the water, below the average, and it is going to be carried out the pollination at there. म्हणजे pollination occur below the surface of water. Here pollen grains are heavier than water. So pollen grains sinks into the down. And it will be caught by the stigma of female flower. In Zostera, seagrass, the pollen grains are long, ribbon-like, and without exile. Let's move to the next one. Epihydrophily. So epihydrophily means what? The pollen grains float on the water surface and reach the stigma of female flower. We call this as an epihydrophily. So epi means above. So pollen grains are released in the submerged state. It will becomes up, float on the water, and where it is going to be reached to the stigma. त्याला आपण म्हणतो epihydrophily. मग यामध्ये काय काय येतं बघा. So freshwater aquatic plant in which female flower reach the water surface temporarily to ensure pollination. म्हणजे फ्लॉवर जे आहे ते वरती येतात टेम्पररी, ensure the release of pollination. मग मेल फ्लॉवर जे आहेत which are float on the water surface and these pollen grains are came in contact with the pollen grains are came in contact with the female water. Specific gravity of pollen grains is equal to water, so that pollen grains are float on the water surface. Some aquatic plants are anemophilous, त्यामध्ये पोटामोंगेटॉन आहे आणि होलाग्रिस आहे. सो सम aquatic plants are anthimophilous, की ज्यामध्ये लोटस आहे, वॉटर एसिंट आहे आणि वॉटर लिली आहे. ओके, सो आय थिंक we're going to be okay. Here we are having the particular structure of Felsenaria plant. So what we going to be observe at here? This is the male flower. This is the water level, and here we are having female flower. So whenever it is going to be released as pollen grains, these pollen grains come up to the surface, and in the surface it will be came in contact with female flower, and it will be leads the pollination. Let's move. Okay, 5 मिनिटं राहिलेले आहेत. ओके, आपल्याला नेक्स्ट पार्ट पाहायचा आहे बायोटिक एजंट. मी डिस्कनेक्ट करतोय, इमिजिएटली जॉईन करा. ओके, इमिजिएटली जॉईन करा. थँक्यू.